From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on October 8th, 2024. Hello, Micronauts, and welcome to our Quality Quorum for today. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 62nd episode of Matters Microbial. I genuinely appreciate every listener and viewer of this podcast. Thank you all so very much. My Bio 350 Micronauts this week were proud to be in our course, as you can see with this set of Bio 350 plates with colorful microbes. They also collected seawater samples and plated dilutions on two types of media, Seawater Complete and LBS, to begin a marine microbiological safari. As seen from this typical plate, there are a wide variety of colony types, phenotypes, to be seen. One phenotype of interest to my research group are marine bacteria that degrade the agar that solidifies the nutrient medium. Since agar is made from seaweed, this is not a surprise. As seen here, tilting the plate can show the pits that such bacterial colonies leave behind as they digest the agar around them. There are many types of bacterial pitters, and yes, we call this investigation a pitter party. Here is an image of a light-colored pitter, and here is an image of a pigmented or dark-colored pitter. We're collecting the various types of bacterial pitters for further analysis. Now, some seawater samples also include bioluminescent bacteria, as seen in this video. So beautiful. Some marine colonies secrete a pigment that fluoresces under ultraviolet light, presumably as a siderophore to collect iron, as seen in this video. Students love that one. And some plates showed a variety of colony phenotypes. As seen in this video, you can see a pitter, a slightly fluorescent colony, and one colony that appears to fluoresce red. So Micronauts select interesting candidates they observe for further analysis. So it really is a marine microbiological safari. Moving on to today's topic, we have discussed bacteriophages, viruses of bacteria, several times in podcast sessions. Here is a 3D printed image of a T-even bacteriophage, and it really does look like something out of science fiction. It is true that when bacteriophages infect their host cells, they become pirates, taking over the metabolism of the host, turning it into a factory for making more viruses. The final step of this genetic and biochemical program is the release of the progeny phages from the host cell through a process called lysis. This final step is under intense study, as learning how proteins can lyse bacterial cells could have applications to medical science. So I'm very, very pleased to introduce the always busy and always cordial Dr. Jolene Ramsey to discuss the work that she and her coworkers and colleagues do in the study of bacteriophage biology, with an emphasis on that final step. Welcome to the Quality Quorum, Jolene.
Hi, Mark. It's great to be here.
I'm really looking forward to this one. I don't know if you know this, but I did my PhD work on bacteriophages of Rhizobium.
Yeah, that's a really good start for any microbial scientist and just scientists in general.
Exactly right. I thought it might be a good way to start. You might discuss your path to the microbial sciences, because I often say that students and other interested people believe there's only one path to being a microbiologist, and this isn't the case.
Yeah, I'd love to talk about that because it really started out with several mentors of mine. When I entered college, I didn't have a good clue, like many students, what I was going to do with my life. And I took a biology course because it was part of the general education requirements. I enjoyed it a lot, and my professors recruited me to take the second semester of intro biology just because. And I thought, well, I guess I have time. This is only my first year. And throughout that second semester, my professors really managed to kind of reel me in and get me to change to a biology major. And I was working at the library at the time, and my one of my bosses was married to a biology professor, and she suggested to me that I might think about working in his research lab. So I did start doing that. I worked with Dr. Gauss, and his wife Nancy was the one who had suggested it. I spent a lot of time with him on actually bacteriophage studies because that was what he studied. We were trying to clone some genes and grow things up in the lab. And being at a really small undergraduate college, there were only about 2,300 students in the university at the time. And there was really just me and Dr. Gauss in the lab. And I think those times of learning and discovering and kind of running my own little research project there were really what got me into the entire scientific research enterprise. And then, of course, I went on to do more research experiences at other universities with my doctoral work that were not exactly on bacteriophage. But when I went on to do my independent training as a postdoc, as well as when I established my lab, I continued with phage work because that was really what got me the most interested. And I love everything about how you can use them for doing research.
I think it's important for people to understand that there are such a variety of different bacterial viruses out there. We're used to the idea of the diversity of viral types in general, but even with the viruses that attack bacteria, there's a huge diversity. Some of those viruses become tools. That's certainly been my experience. And others are just interesting in their own rights. So it's a wonderful place to start. I believe you also hit the nail on the metaphorical head when you mentioned the value of mentorship, someone who takes an interest and encourages you personally. And that worked for me, although I did not go to a small school. I was still able to get some mentoring in, even at UCLA. But as I have spent my career at small undergraduate institutions, what you say resonates with me. So I think that maybe after this, you had mentioned to me earlier that you wanted to kind of introduce the idea of, and please forgive this, a bacteriophageology to the listeners and viewers.
This is a question that I love explaining to people. What are bacteriophages, the viruses that infect and kill bacteria? They are everywhere. First of all, they are in every environment where bacteria are, and bacteria are literally everywhere. And they are in different amounts in those environments, but they tend to outnumber their hosts. Estimates say about 10 to 1. And they are of all types, as you said. In what that means is they have different kinds of genomes. So the genetic material that they use to replicate themselves can be RNA or DNA. Now, the ones that have had the most study done on them have a double-stranded DNA genome. And those are really of a very nice charismatic character because when you look at them under an electron microscope, you can see that they have really interesting shapes. They look like, some people describe it as a moonlander. Some people describe it maybe as a spider. What we describe it as having a head or a capsid that encapsulates their genetic material, and then usually some sort of tail. And that tail can be short or long, and the longer tails can be flexible or rigid. And the rigid tails actually can contract almost like a syringe. So those double-stranded DNA phages have been highly studied. They're easily isolated in the lab, but there are also some very abundant viruses that do not have those shapes, that have kind of more what look like round shapes, but that are really actually beautiful geometric arrangements of the proteins that make up that capsid head. And then there are some that are even more different shapes, some of them that are what we call filamentous. And those can be long, kind of rod or pencil-like. And then there are some that are just pleomorphic or have many different kinds of shapes and maybe don't have a single type of shape. And all of those differences between them are really some of the most interesting things to study about them. How do you get those different shapes? And how do those different shapes impact how they interact with their environment? And even with these different kinds of shapes, they do share some commonalities in the way that they infect their host cell. So I love to describe this as stages. Initially, the phage will have to encounter its host in the environment. And this generally happens by random chance. They bump into each other. And if that initial bump has a specific interaction between the phage and some molecule on the surface of the host cell, then that can then turn into an infection where the phage will recognize, and by recognize, I mean have some conformational rearrangements that allow it to inject or eject its genomic material into the cell. And after that initial stage of infection, the entry stage, which actually can be quite fraught because you can imagine that getting a piece of nucleic acid across a lipid barrier and all of the surface molecules that are protecting that cell is no small feat, actually. Once it gets inside, it has to establish its dominance, you could say, because that host cell usually is not going to be leaving itself vulnerable, having been encountering many infection cycles in its population's genetic past. So there are many defenses that host cells can deploy to prevent the phage from infecting. Those take the form of enzymes that will cut up the DNA or block its replication. And those are the kinds of things that, as we studied bacteriophage, turned into really important scientific tools that we use in the lab. They can also take the form of things that will prevent the cell from functioning. Let's say the phage has gotten in, it's starting to try to take over those cell functions, like you said, being a pirate, it's taking over, establishing that this is now my ship. The cell can shut down all of those normal processes that the phage needs to be able to make more pieces of itself and assemble more virions. And sometimes the phage includes defenses. Sometimes it's just speedy. Sometimes it has specific defenses against those defenses. We call these anti-defense genes. And once the phage has won, if it has, which is not always the case, then it will begin to replicate its genomic material. It will make more of the proteins that are used for encapsulating that genomic material. And it can do lots of interesting things along the way, because as the host cell is either shutting itself down or the host cell might be different from maybe the last one that the phage encountered, sometimes they need to ramp up the production of a particular nutrient or a particular subcellular component that the phage will use to build itself. So there's lots of interesting different kinds of properties that the host cell can take on as a phage has infected it. And then at the end, then the phage has to get back out through all those layers that it crossed on its way in. And since it's coming from a different direction, it takes some different strategies. And that's really where my lab has focused a lot of its time studying the molecular processes that phage use to lyse or burst open the host cell at the end of the infection cycle. Now, one thing that I didn't mention is that sometimes the phage gets in and it decides, instead of immediately replicating, it will settle in and take advantage of the cushy situation it has found, where it is protected from outside forces that might try to prevent it from having a new infection. And it will sometimes, what we describe, form a partnership with that host cell. And it will kind of lay low and covertly become part of the host cell and just go along for the ride as the host cell grows and replicates. And we call these integration, or these events where the phage will become part of the host cell's replication cycle, lysogeny. The phage becomes what we call a prophage, almost like a pre-phage or something that could become a phage. And then it will remain in that state until some signal induces it to realize that the external environment isn't going to make this a good place to stay, and it's time to abandon ship. And in that case, it will reactivate and begin its replication and get towards that lysis step at the end.
Kind of like hoisting the Jolly Roger if we're following the piracy metaphor.
Yes, exactly.
Do you have some thoughts about how you can construct something as complex as a phage, a bacteriophage virion, does that occur step by step? Are there different mechanisms taking place? Are they using kind of host structures to do so?
Yeah. So the process of phage assembly, or sometimes we call it morphogenesis, is really fun. It occurs in stages depending on how complex the virion itself is. And the first thing that we usually think of is the assembly of the capsid head. And that's typically, well, it kind of actually depends. There's two ways it can happen. One way is the protein itself that forms that capsid will self assemble. Actually, it is really a self assembly. And actually, I have here I'll show for your viewers. This is a 3D printed version of a representation of a virus head. And I have here several pentamers, and these pentamers have magnets on them. And the reason there's magnets is because that represents the protein-protein interactions between the capsid that occurs spontaneously inside the cell. That's why we call it self-assembly. So I use this as a teaching tool and an outreach tool. You can crush the model, and then you can put it into a box and then shake it up, and it will self-assemble just like happens inside the cell. And if there's a lot of other stuff that is in the cell, then that's going to kind of maybe make the process a little bit longer. But they do self-assemble. And usually then for a lot of the phages that I've been talking about, the nucleic acid is then pumped inside the cell, inside the head with a molecular machine, and then that's capped off. And then the phage tail, which also spontaneously assembled by protein-protein interactions that are specific inside the cell, that gets docked on. And then any other decoration proteins are also added. So it really is an incredible self-assembly process that we can do in a test tube if you have all of the right components, because they will assemble in the correct order. Sometimes, especially with eukaryotic viruses, this has been seen, but there are some phages where we think this happens. The nucleic acid nucleates that process of assembly, and
Right.
you get the proteins will interact with the nucleic acid and then form the shape of the final phage.
Thanks very much. I really want one of those self-assembling models. I've talked a lot to my wife, Dr. Quinn, about that one. She does a lot of 3D printing, but that's a lot of magnets. That's the issue.
That's the tricky part, is gluing in the magnets correctly.
Yeah. It's really fun to hear these things, because I have also really enjoyed bacteriophages, though I didn't really continue to work in the field. And I remember when I was a graduate student and we were using this large T-even bacteriophage as a taxi cab for moving genes around through generalized transduction. And I was curious about the genome of this bacteriophage, and I discovered that it had shifted it hypermethylated its genome to prevent restriction enzymes from getting it. So, but some of the time it just was literally missing the various restriction enzyme recognition sites. For listeners and viewers, restriction enzymes are a natural thing that many bacteria make, and they're used to fight incoming DNA of various forms. They recognize a specific sequence and they cleave there. Mostly we use them as tools nowadays, but of course, microbes invented that a long time before. And as a result, one way to get around that is to methylate the DNA, which prevents that recognition. However, I was able to show in these bacteriophage that they're simply missing some of those sites, which is amazing to me, as well as the hypermethylation. So even in this tiny supposed pathogen or predator of a bacterium, there's so much diversity of approach.
That's right. Yeah, those defenses such as restriction enzymes and CRISPR systems, they are defended against by, as you mentioned, anti-restriction mechanisms, including modification of the sequence of the nucleic acid or production of a protein that will block its activity.
It really is like the Red Queen, running ever faster to stay in the same place. And I keep coming back to the role of evolution in everything. And it's a good thing to remember. I mean, I don't want to make this, my own memories, but it's so delightful to hear this aspect of things.
Mark, I think the thing is that phage biology is really what started molecular biology in the first place. There were studies of just how they worked, and people use that for training. They use that not just for training, though, also for discovering how the cell works and applying those discoveries to produce biotechnology tools that were critical for the researchers to move on to study other organisms and that we continue to use today. So phage continue to produce the study of phage continue to produce very important uh, discoveries such as the CRISPR revolution.
Yes.
And there are anti-CRISPRs that phage produce that allow us to have kind of control handles on CRISPR systems. And these wouldn't have come about if there weren't this kind of tit for tat going back and forth with phage. And we can still use phage to discover the novel things that lie in the microbial secrets that we haven't yet discovered. So I think that even though it might seem like sometimes we're studying these very obscure things, it turns out that they really teach us important parts of how cells work, as well as provide us with tools that we can use for our society.
I mean, some might say obscure, but I prefer ubiquitous because they really are ubiquitous. Do you know the story? And I have to go look up his name, of the German physicist who first did an electron micrograph of bacteriophages?
I don't think I know that story.
And I got it in the History of Molecular Biology, a wonderful book I'll put a link to in the show notes. But he supposedly said, please forgive the accent, mein Gott, they have tails.
Mm-hmm. Yes,
And
yes.
I mean, to me, I love the idea of, the first person to see something. I mean, that's the purest expression of science ever. But your own work that your interest lies in is that final step of release from the host cell.
That's right. Yeah. So phage lysis is it's something that is interesting because they don't all do it the same way. So it turns out that there are multiple strategies for crossing those physical barriers that are trapping newly formed phages inside of a host cell. And we generally divide these into three, I would say, different strategies. One strategy that we study, it uses multiple gene products, multiple proteins to target each of the layers of the host cell. There are phage that make one gene product that inhibits an essential cell process to build that structure. And then a third category, I would say, are the ones that don't actually really completely lyse the host cell to get out. Those are the kind that are using the host cell as a vehicle to bud or extrude is the word that we use, new phage particles. And that's because they use some of the material of the host cell envelope in their actual virion. So in terms of the multi-gene lysis process, this is something where it's going to be dependent on the specifics of the host cell that the phage is in. Now, most host cells are going to have a membrane layer or a lipid fatty layer, and then they're going to have a rigid layer that helps them keep their shape. That's called peptidoglycan. This is a molecule. It's really a very large molecule that is cross-linking multiple smaller components of peptides as well as sugars. So it's a composite of both of those. And that is something that has, it's a meshwork that holds the cell shape. And so the meshwork openings are too small for the phage to get through. And that's why they have to degrade that layer usually, or prevent it from being as rigid as it is. And then some cells have extra layers outside of that peptidoglycan. Sometimes it's an outer membrane layer. Sometimes it is a waxy outer layer that is a mycobacteria have a more modified type of layer. And then sometimes they have other protein layers that are out there, capsules and so forth. And so the phage produce specific proteins for each of those layers, depending on which host cell they typically infect. Now, in my lab, we study gram-negative bacteria that have two membranes and a peptidoglycan layer in the middle. And the first membrane is targeted in a really interesting fashion. The phage makes a membrane protein that goes into the membrane, and it hangs out there harmlessly until it reaches a specific concentration. And when it reaches that concentration, the critical threshold concentration, we call it, it will rearrange itself and make these giant holes. And those giant holes now have broken down that membrane barrier and start to mix the middle compartment and the inner compartment of the cell. And that also is the step that kills the cell. And I think this is a really interesting aspect of the way that the phage gets out. It kills the cell before it actually gets out. So if it doesn't have all of the proteins necessary to degrade each of the layers, then it's actually trapped inside of a dead cell, which is not a good place to be for a phage. But usually after that hole is formed by the whole-in protein, there's another enzyme that is released and will degrade the peptidoglycan layer, which is called an endolysin. And then the last part is the use of some proteins that will destabilize the last membrane. Sometimes these are called spannins. Well, in the majority of cases, they're called spannins. And we also study some other versions of proteins that can do that called the disruptin. And I do have a diagram of this in a microscope form, as well as in cartoon form that I have put for your viewers who might be seeing a video. And you can see how it's a very quick process because the start of the process, the cell looks normal in the microscope, but very quickly, in less than a second usually, the cell will have all three of those layers breached and it will burst open to release those phages that were on the inside. So in our lab, we have been studying the actual mechanisms that the different proteins in those three stages use. Sometimes the holes are bigger than other times. We've found that there are some phage that actually don't have a protein that makes a hole. They have some other trick to get around that inner membrane. There are variations, as I said, in the kinds of proteins that target that outer membrane. And we're interested in some similarities that we've found between those proteins and the proteins that the human innate immune system produces to defend itself against bacteria. And I think that's another reason that a lot of people find what we do to be of great interest, because we're studying the proteins that kill bacteria. And as you know, Mark, not all bacteria are problems for people in the clinic, but some of them are. And there are also sometimes problems in cases where like maybe a bacteria will normally form a biofilm. And if it does that inside of a pipe and that builds up, then that's a problem. Or if it does that on the space station where we need to keep water flowing, that's also a problem. So the idea that we can use some of these proteins and understanding how they work to treat and get rid of some of those biofilms in places where we don't want them in our built environment is very attractive.
Do you mind if I ask an odd question?
Oh, please, go ahead.
So I know that when I'm teaching microbiology, there are many things that eukaryotic cells make to try and defend against them. And they have funny names like perforins, because the idea is they poke holes in things. And in fact, there are a number of these sorts of things going on. Are those perforins that eukaryotic cells, including us, make, are they related to the wholins that you're talking about?
Yeah, it's a great question. Actually, I've asked myself that as well, because there has been quite a boom of research that is showing that there are relationships and similarities in many ways between the proteins that are defense proteins in bacteria and the proteins that eukaryotes like ourselves make to defend ourselves against bacteria. And I would say that we know that there are similarities between the whole-in protein that the phage makes and the proteins that eukaryotic cells use to turn on apoptosis. There are several other kinds of proteins that eukaryotes make to produce holes in bacterial membranes. Another one is the Mac attack complex. So I do think that in some ways they are similar. The themes are quite similar. It's just that the direction and the control that they're coming from is different.
I also was curious about something that I saw myself when you have too many bacteriophage attacking the same cell, you can just pop those cells instantly.
Hmm. Yeah, so this is something that's of great interest to the field. Um, Part of the reason that that occurs is you can imagine a host cell, and you have one bacteriophage that will adhere to the outside, and it needs to cross those layers to deliver its genome. Now, if it's just one and there's a hole made in the membrane, then that can be relatively quickly repaired by the host cell and they can recover. But there is a little bit of leakage that occurs during that. And it kind of depends on the sort of phage that it's infecting, depending on if it has a tail or if it's using some bacterial structure to get across the host cell membranes. So if it's poking a hole and that takes some time to repair, then you can imagine that if you have one hole, you can repair it. But if you have 100 holes or 1,000 holes at once, the repair process is not fast enough. So we call that lysis from without.
Doubt.
And that is definitely possible when you add too many phage. And actually, I wouldn't even necessarily call it too many. It's just there's a high number. It's something that most people observe without even realizing it when they are doing something we call titering, which is counting the number of phage in a particular sample on an agar plate. You take a highly concentrated sample and then you make dilutions of it until it is a lower concentration. Then you spot each of those onto a host, a thin layer of host cells, and wherever the phage is, that will cause killing. And if you have tons of phage, then you can get killing, even though the phage doesn't usually infect and replicate in that host.
It's amazing stuff. This is taking me back to graduate school, which and I got to tell you, one of the things that you know and I know, but I want to make sure that viewers and listeners know, is that bacteriophage are really interesting in that they stop killing when the cell stops living or growing, more accurately, for the most part. There are some
Yes,
exceptions, but it's important to know.
that's right. So many phage can only successfully replicate in growing host cells. And that's because they rely on all of the processes that the host uses normally to grow and divide. So they can't replicate without the ability to make new proteins and make new DNA or RNA. And so if the host cell has shut those things off and the phage can't reactivate it, then it's stuck inside that cell. It's cool that there are some phage who have manipulated the host cell processes to reactivate. Yeah.
It's so interesting when I teach microbiology, students expect things to be very linear and hierarchical and everything to make perfect sense. And I have to say that, and I talk about this, that there's such a thing, and I call it coli centricity, where people think that all bacteria are like E. coli, and in fact, not all E. coli are like E. coli.
That's
Um,
right. That's right.
and it's important to remember the diversity of the world around. I mean, even if you look at, for example, and I'm showing the 3D printed T even bacteriophages, and I could never print this on my machine, but this has to be assembled inside the host cell
Hmm.
before release. And to me, and that's why some people early on kind of used it as a model for developmental biology, simply because they have to be assembled in the correct order.
Hmm.
But this is one of the things that I really try and bring across to students and other interested folk, is that it's a delicate ballet all the time inside all of our cells.
Yes, that's right. And the study of this is something that is really quite suitable for students to use to start to understand both a little bit of the abundance of these entities in the environment, as well as how to do the research in the lab. And, kind of returning to that idea of reactivating the host cell, I have a graduate student who joined my lab this year, and she started in a really fantastic educational program called Phages that is run through the Pittsburgh Bacteriophage Institute and funded by Howard Hughes Medical Institute. And she started as a 16-year-old high schooler, pulling soil out of the environment and looking for those clearings on a layer of host cells that we call plaques. And she found some, and she then started to do some research on this one phage that, after leaving that plate on the bench for maybe a little longer than we normally would have, noticed that the plaques just kept growing and they never stopped, even after more than 40 days. And so this is just a really cool way for students to discover interesting things. She went on to do her thesis work on that phage as an undergraduate, and now here we continue to do these phage discovery efforts. For your viewers, I have put a little outline figure of what we call the phage hunt, where you start with samples that are from the environment, and you mix those with the bacteria that you're interested in finding phage for. And those environmental samples that we collect are collected based on where that bacteria lives. So if we're looking at a bacterium that is normally found in the soil, then we will collect soil samples to look for phages that infect it. If it's a marine bacterium, we'll collect marine samples. If it's a bacterium that's associated with human infections, then we will look where humans, many human materials are collected in one place, and that is the sewage. So students will take those into the lab, look for those really wonderful plaques that we can see on the plate, amplify that in the lab, and then they get to see the morphology of their phage by electron microscopy, which is one of the favorite days in either a class or a research project that's doing this. And then we take out the nucleic acid from that phage and do some analysis on it, both to understand it on its own and maybe to kind of tease apart the behaviors that we have observed in the lab, as well as to relate it to other phages that are in the environment. And I think that this is another aspect that is a really exciting and fun thing to do. So while I greatly enjoy the entire phage hunt process, it turns out that most of the phages in the environment have never been cultured in the lab that way, just like most of the microbes in the environment are not cultured in the lab. So it turns out that we can get lots of sequences from the environment that are phage sequences, and we don't always know which host they infect, but we can learn a lot about them just from their sequence. And this is another area that I think is a fun and informative place for students as well as microbial enthusiasts to get involved. And I have one more figure there that viewers can see. We have depicted here a person with a flask, and in that flask is their newly isolated phage, and they're on a journey now to figure out what can we discover about it from its nucleic acid. How does it relate to all of the other phages in the environment and that we know about? Or is it completely new? Because it turns out that many of them are very different from what we have seen before. So it's really a journey. A postdoc of mine in the lab, she just published an article on this process of classifying phage and how you do that both on the morphology side of things as well as their infection cycle, understanding what type of infection cycle they choose, whether that's a hunker down and wait for later while the going is good or the let's get out of here as quick as we can strategy. And then understanding where it stands in terms of its classification officially, and then getting some more distant evolutionary relationships out of it. So we call that the supergroup classification. And this is really meant to be an introduction to the newbies on how phage classification works. I think that it's a lot of fun to find out what's new and what's similar to what's out there. You'd be surprised. I've been surprised so many times to see that a phage that we isolate here in Texas, where I am, is super similar to phage that are isolated in countries around the world because there's just a lot of genetic exchange going on around the world.
You've talked about so many things that are of great interest to me. And I just want to, I took a couple of notes here and I wanted to tell you. So I wanted listeners and viewers to understand that where a single phage virion lands under the assay you're talking about
Hmm.
will begin to replicate locally, infecting cells right next to it until those cells stop growing, leaving this hole, which we call a plaque. Those plaques are actually the result, again, of a single bacteriophage virion, a single phage particle.
Hmm.
And because of that, they stop after a certain point. Now, you mentioned a phage plaque that continues to grow, and I had to smile with great excitement because an organism that I have tried to study for many years, Delavibrio bacteriovorus, a
Mm.
bacterium that lives on other bacteria, forms plaques that continue to grow because Delavibrio
Ah,
can grow on dead cells.
okay.
So I am going to be reading about this particular finding of your colleague because that is the standard way to look for Delavibrio-like bacteria are plaques that continue to grow.
I see.
And what's interesting to me is that people said, well, there's no, we can't find any cells in there, but they never reported it. I promise
Mm.
you that this is something that can be seen and has been seen that nobody followed up on.
Hmm.
Your colleague, your student was the first.
So I'll tell you, her name is Gilly Brown, and she published her thesis on phage Hasitha from Western Kentucky University. And it turns out that there are interesting other phages that are being reported that can do this as well. We discussed one for a phage named Paridae that infects Pseudomonas. Gilly's phage infects a mycobacteria, I believe.
Mhm.
So this is something that's becoming more recognized as we make more of these observations and kind of delve deeper into their biology.
What's the quote? To see what everyone has seen, but no one has thought.
Mm.
And this is why I tell students, even though we love the very expensive and wonderful machines that some of us have, the most important machine is between the ears of the observer.
Hmm.
And this is what I tell undergraduate students because they often are quite negative about themselves. Your student isn't like that because she knew that what she found is something really remarkable. But I keep telling my students that any one of them, if they look closely, can find things that no one has seen before. And that's the real excitement of science.
I agree. I agree.
Sorry, I was preaching.
That's okay. No, I think that teaching students in a way that they can discover and own is quite powerful.
There's just nothing better than the look on their faces,
Hmm.
when they did it,
Yeah.
not me. And too many times, and this is something that CPhages is really one of the champions of. So many times when students are doing work, it's not really a research experience for them. They're just doing examples of experiments where they
Yeah.
know what's going to happen. So of course they don't view that in a terribly excited fashion. But when they find something that other people haven't thought about, this is the value of these of cure-based classroom experience. Wow.
Yeah.
And it really makes a difference. It actually creates scientists no matter what they end up doing later on for a career.
I agree. And I think that the excitement as well of sharing that discovery with other people is also something that really makes an impact on students. And it helps them see that their education is not just for a grade that their professor is going to give them, but that it's really something that matters to the rest of the world. And that's the subject of the other class I wanted to tell you about, which is
Please.
my CACAO class. So this is a really exciting project that I have worked on since I was a postdoc. I started as part of the team under a wonderful educator and researcher named Jim Hu. And I continue it now with his partner in crime, Debbie Siegeley. And it's a course where we teach students about reading the primary literature, and we do it in a way that is practical and has real world applications. So you can imagine this way. The research problem is that there are more than 37 million articles indexed in PubMed that are not machine readable and certainly not readable all by one person. But the information that's in them has taxpayer-funded potential solutions to our biological questions. So we want that to be accessible. And then from an education standpoint, undergraduates need to learn to read the primary literature with a critical eye. So our solution is to have an authentic research course called CACAO for creating annotations through critical analysis of original research. And I have a little figure of this for your viewers if they want to follow along. And this is where students read the primary literature and they extract the information through an activity we call biocuration. They extract it in a way that it can be input in a regular formatted way into databases online that both humans and machines can read as something called an annotation. Those annotations get peer-reviewed by instructors in the course as well as other classmates. And this is the part where we have gamified it. So the students work in teams and they put them into an online database where other students can see and they can make corrections. And if they make corrections, then they can earn the points for that annotation rather than the original inputter earning the points. And after those are completely peer-reviewed and correct, they get pulled out of our CACAO game system, which is a website, and we put that into the public databases. In our case, we're using the gene ontology, which is an excellent way to express biological information about genes, processes, locations, and functions in the cell in a way that scientists can use it for analyses of large data sets. And that is available to scientists worldwide that they use to do the analysis of those large data sets. We do this locally with teams of students, and students earn points for making completely correct annotations. And their standing in the competition also earns them chocolate prizes. That's why it's called CACAO. The course is an intercollegiate competition, so we can generate robust peer review, which is another thing that teaches them about how scientific processes work. And in the end, so this course started long before I was involved, back around 2010. Since then, students have generated over 5,000 original annotations from the literature, nearly half of which, this is of interest to you, are to microbes, both viruses and bacteria, and archaea, actually. And that's really been an important research contribution because very few of the annotations that were available before we started were to microbes. So we have been able to contribute a resource for scientists to use when they're doing their analyses of large data sets. For any scientists that are listening, this is the kind of thing that would be used for analyzing mass spec or a giant RNA-seq type of data set where you've looked at all of the RNAs or proteins present in different conditions and you want to know what are they involved in, what kinds of pathways and functions do they have. And so you can group them by using these gene ontology annotations that our students are making in the CACAO course. So I love this course because students find it to be invigorating in the competition aspect, but also real world and practical in that they're really contributing to the scientific enterprise in a way that other scientists haven't been able to do due to lack of time. But we're doing it in a format where the students get academic credit for it, as well as chocolate.
I really love this idea, and I am going to put links to your class, and I know you have a publication on it, and there'll be a link to that as well. And I want to emphasize that one of the most difficult things, and I'm at a small undergraduate institution, so I don't have graduate students or postdocs or that kind of thing, but you don't have the same kind of culture at this sort of, that's kind of a pun, isn't it? That kind of a culture at a small liberal arts college. But one of the big challenges is getting students to read the literature. So I found what you are talking about to be very intriguing and interesting because of what we talked about before. There's no substitute for actually doing some level of ownership on the research that you're doing. So a cure-based program where you're actually doing research instead of just a series of exercises really does invigorate students. You've taken it farther now in teaching what I would call a certain bioinformatics set of skills in a particular gamified format that'll have the same kind of effect that helps people learn how to read literature carefully. I
Hmm.
think sometimes students believe that papers get handed down on stone tablets. And in fact, that's not how papers are written. That's not how they're received. And I think it's really important, especially nowadays, for people to understand how science is done.
Yeah.
And this is at every level what you talked about really sings to me. So I want to thank you for that.
Yeah, well, it's been a collaboration. This is a really great project, and we always invite more participants to have teams that could even participate just for a round of annotation, because I think that it really generates some additional excitement as well as some new topics that are going to be added as annotations to the Go database.
Well, this is wonderful. I really thank you for sharing it. Do you have, and this is going to be a way out there question, do you have a view of where your research activities are going to be in 5 years' time or in the future? Do you have like a goal, or are you letting the research take you wherever it takes you?
We definitely have some ideas of where we're going based on the work that we're doing now. Kind of wrapping all of these things together, we're really interested in discovering or having a better idea of the diversity of mechanisms that bacteriophages can use to lyse open host cells. We want to understand the differences in how the different kinds of proteins work, but also we want to really dig into the regulatory mechanisms that phage are using to determine when they lyse the host cell. And I think that this is important to keep in mind because phage lysis occurs in every environment where an infection is happening, but the environment is not always the same. And phage can actually, in a way, listen to what's going on outside the host cell to determine if it's really a good environment or a favorable environment for them to escape into. And if it's not, then they can delay their lysis process. And that ability and the ability to harness or control when a phage lyses a cell is, I think, kind of where we'd like to be going. We want to understand what are the kinds of signals that can impact the timing of phage lysis, and can we listen in, and can we even jam the communications to determine the timing of lysis, and can we use that to understand the ecological dynamics of phages in different environments, in the gut, in the ocean, or in maybe even some places where bacteria are in the clinic as well. That way we can harness the power of the very well tuned lysis strategies that phage have come up with to both understand how they work, but to then potentially manipulate that process in environmental processes that we care about.
So fascinating. And one thing I was thinking about as you were talking, do you remember you were discussing about that initial attachment event?
Yes.
And many bacteriophages have a specific receptor. Bacteriophage lambda uses the maltose receptor on E. coli, that kind of thing.
Yes.
I remember some work being done in chemostats many years ago where they controlled for the number of host cells.
Hmm.
And they were able to, and I use this term advisedly, do evolution in the chemostat. And they were able to show under situations where you didn't have many host cells, you've got better attachment on the more successful viruses that evolved under those conditions. And what I really worry about to some degree when we use the workhorses is how similar they are or dissimilar from things that are freshly isolated. So I applaud you for looking at freshly isolated bacteriophages as well as the ones that we've cultivated in the lab for some time.
Yeah, there's a lot of new things to be discovered from isolated bacteriophage that haven't been studied yet. But I also think that there are many things that we haven't yet uncovered from the ones that have been well studied. So adding in and mixing those things together is really going to help us harness the power of all the tools that we've developed, but not just focus on things that we've already seen.
They've been around for a long time. They have a lot to teach us. I thought I would, and I have to look carefully at your face when I'm telling you this story. When I used to work in a biotech company and I worked with Xanthomonas campestris, which makes xanthan gum, which is used in the food and actually the oil industry, both, don't ask. And what's interesting is that from time to time, because they would not listen to people like me, they would get phage outbreaks in the giant fermenters. I want you to imagine a 100,000 gallon fermenter turning to froth, because that would happen. And then we would have to isolate ourselves and make bacteriophage resistant versions of the strain and make sure that they hadn't lost their production capacity.
Yes.
And it was a continual battle, because I said evolution always wins. Darwin wins, always. And you can't view any of these things as reagents. Even when I talk about a bacteriophage, please forgive me, purists. I view them as part of the living world. So you're going to have evolutionary effects.
Yeah, I think that the way that I've met people who are not really interested in phage most commonly is when they have an event like that. Usually not quite a 100,000 gallon problem like that, but in the food industry and in what you've described, phage infections like that are not the kind of thing that you want to see. So knowing how to shut them down or prevent them is going to be helpful, but it's not going to be a permanent solution.
That's just amazing. So Jolene, I have a question for listeners and viewers that I think that might be occurring to them. Many times when we talk about bacterial viruses, people wonder how the study of that differs from the study of viruses that cause diseases that we're more familiar with, such as influenza or SARS-CoV-2.
Sure. So it turns out that a lot of the kinds of approaches that we take to understanding the infection cycle of a phage and of a virus that infects a eukaryote like humans are very similar, because their infection cycles are to encounter a host cell that it specifically can infect, get inside, take over the host cell, assemble itself, and then get out in sometimes a lysis process and sometimes not a lysis process. However, the structure of the host cell is quite different, and the machinery inside the host cell is quite different. So the reagents that we would use and the timeline that we would be studying it on would be longer. The reagents would be specific to that kind of eukaryotic host. But, that's actually similar in phage research as well. So if I want to be studying a phage that infects a gram-negative cell versus a gram-positive bacterial cell, the reagents are going to be different. And in the same case for viruses that infect archaea. So the specifics of the studies are going to be dependent on who the host cell is. And there are definitely more proteins and more cellular structures involved in a eukaryotic host. But the basic principles of viral infection are quite similar.
In several episodes of Matters Microbial, we've discussed the role of communities of microbes doing things together in sort of an emergent property. Are there similar sorts of things going on with viral communities?
That's a really interesting question. So some people have been studying this idea of phage-phage communication. Certainly anywhere that there's a microbial community, there is a phage community as well, sometimes called the phageome. That phage community is going to be composed of the phage that infect the bacteria and other cells that are there. And how they communicate with each other is not your standard type of communication, although there is some suggestion that there can be small molecule communication between infected cells. So the virions themselves, that are the intracellular transport complexes that move the genome from cell to cell aren't necessarily going to be communicating with each other, but an infected cell can send out a signal that's detected by another cell because it's
Mhm.
infected by a phage. So we do think that there's going to be some impact of those phage community interactions based on what they're producing when they infect the cell.
I'm going to hopefully have on the show some people who are in part of a field that's emerging called sociovirology, which fascinates me, the idea of it. So I look forward to that. Now, you've talked a little bit about, and I'm going to just generally call them lytic factors. That is to say, the enzymes being made that lead to lysis of the cell. Are there multiple or simple controls on how they're being made? Because I'm wondering about concentrations and if that's modified by the environment.
Yeah, so the proteins that the phage makes these proteins, and it has them programmed to be made last for the most part. And then they'll be made and they'll start building up slowly. And when I say slowly, I mean it depends on the speed at which the host is growing. So if it's a fast growing E. coli host, then these can build up within a matter of just 10 or 15 minutes to a concentration that it can completely lyse open a complete population of the host. And if it's a slower growing host, or if the phage has detected some sort of signal that maybe it's not quite yet time for it to lyse, then it can put kind of a delay, a hold on that lysis process, even though all of the proteins are there in concentrations sufficient to lyse the cell. And then it can release that hold and lyse the cell when it's ready.
That reminds me, Jolene. Now, I may have this not entirely correct, but Vibrio natringes is a marine bacterium that divides very quickly.
hmm
I want to say 11 minutes per generation.
I think the fastest that has been measured for it was 8 minutes, which is crazy.
I agree completely. Obviously, there have to be bacteriophage that go after it.
There sure are.
Are they as rapid in their development?
I wish we knew the answer to that question. There have been quite a few people becoming very interested in that Vibrio natrigens as a biotechnology tool. So there have been more isolations of natrigens phages. We isolated one that we was one of my first students in the lab. He isolated one, and we called it friendly, and that was in contrast to another one that he was trying to isolate that was called problem phage. Anyways, so yes, the phage will replicate at the pace that the host is replicating. So if at optimal growing conditions, then that would be ridiculously fast.
It's amazing. By the way, on that topic, I heard a rumor. I don't know if it's true. You were talking about the names of bacteriophages that people have isolated. I heard a rumor that the C-phage folk had to forbid the use of Nicolas Cage in the names of any phages that had been isolated. I don't know that that's true, but I like Nicolas Cage for being over the top, and phages are over the top too.
I wouldn't be surprised. They come up with some very creative names, but also popular things tend to pop up over and over. And it's helpful if you have some unique names. And students are so creative that they come up with really great things to call their phage.
May I ask you a question before we close out this wonderful session?
Sure.
Is there one event that sold you on being a microbial scientist?
Yeah, I have to go back to how I started, and that is with Dr. Gauss in the microbiology research lab at Western State College. We would spend many Friday afternoons when the building that we were in was empty of students talking about the result on my gel or the next idea for an experiment or what it was like when he was a graduate student. And I think that all of those conversations that I had with Dr. Gauss are the thing that really sold me on being a microbiologist. There was just nothing more fascinating to me, and I found most of my classes fascinating than the things that I was learning taking microbiology and doing microbiology research with Dr. Gauss. So I have to really credit him and his passion and persistence at helping me along in my budding microbial career at the time.
There is nothing better than watching a student become successful, and I'm sure he feels that way about you.
I love that he would give me these books, the published Bacteriophage T4 book, and he gave me a paper that had this super interesting title that really just hooked me. I was like, really? So it was called The Rex System of Bacteriophage Lambda, Tolerance and Altruistic Cell Death. And I was like, altruistic cell death. I'm talking about bacteria here. What is going on? So these kinds of very interesting questions just really pulled me in.
So now you understand why I use the expression OMG for
Yes,
overwhelming microbial greatness.
I sure
And,
do, Mark. And
and
I agree.
and WTM for what the microbe? I
Mm.
mean, because it's and so many times when microbiology is taught, and not I'm not talking about you or anyone you know or anyone like that, but so many times it's just very rote, what we used to call the bug parade. But it's such a dynamic, fluid, and wonderful field to be in. There is not a better time to be in. I do things now that I never dreamed I would be able to do when I was in graduate school.
Mm-hmm. Yeah.
So I actually wanted to thank you for this wonderful session. I probably became too enthusiastic, but that's how I feel about this kind of subject. Thank you for the wonderful way that you clearly articulated points of view and solid information. I think this has been a wonderful session. I wish you and yours the very, very best. And thank you again, Jolene.
I would just like to say gracias and thank you, Mark, for what you're doing to spread this kind of information to other people so they can be informed about this wonderful, overwhelming microbial greatness.
You're so welcome. This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode, with wonderful links as always, can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. Dr. Jolene Ramsey is in the biology department of Texas A&M University and an affiliate of the Center for Phage Technology. Many thanks, as always, to David Renata for superb editing and Reber Clark for the wonderfully quirky music that makes me smile every time. I hope that you've all enjoyed being part of our Quality Quorum for today. See you next time on Matters Microbial.